Hello, today we are going to discuss about calcium, phosphate and magnesium metabolism, and it will be a very brief discussion, and I'll be talking about the basics, very basics of this. Myself, Santosh Abraham, working as a specialist registrar in diabetes and endocrinology at Scarborough General Hospital. First, let us talk about calcium. The requirements and absorption of calcium is as follows. So the total calcium in the human body is around 1 to 1.5 kilogram, and 99% of that is in the bone and 1% is in the extracellular fluid. So about only 1% is in the ECF, and adults require about 500 milligram per day, children about 1200 milligram, and it will increase to 1500 milligram per day during pregnancy and lactation, and adults who are above 50 and elderly will require more. And absorption is from the first and second part of duodenum, like folate and iron, and it is absorbed against a concentration gradient, and it requires energy, and it needs a carrier protein and is helped by an ATPase. Factors increasing the absorption are vitamin D, so it induces the synthesis of calbindin in the epithelial cells of intestine. PTH, it increases calcium transport from intestinal cells. Acidity also favors calcium absorption, and lysine and arginine increase calcium absorption. And those which decrease absorption of calcium are phytic acid, oxalates, such as some leafy vegetables are source of oxalates, and they form insoluble calcium oxalates in the intestine. Malabsorption syndromes, and phosphate. High phosphate content will cause precipitation as calcium phosphate and will impair the absorption of calcium. Calpains are an interesting group of proteases. So these are calcium dependent cysteine proteases, and these are present in all the mammals. There are two main isoforms, calpain 1 and 2. They are involved in cell mobility, cell cycle progression, cell fusion in muscle cells, etc. Defective calpain 3 has been implicated in limb girdle muscle atrophy. Calpain 10 is present in susceptibility gene for type 2 diabetes mellitus. Calpain 9 is implicated as a tumor suppressor gene for gastric cancer. And hyperactivity of calpain is implicated in Alzheimer's disease, cataract formation, myocardial ischemia, and incerebral ischemia. The actions of calpains are as follows. It increases intracellular calcium. So any intracellular calcium results in calpain activation. So unregulated proteolysis and irreversible tissue damage results when the calpain is activated. And this leads to degradation of cytoskeleton and plasma membrane. In MI, when there is reperfusion of the ischemic myocardium, there is calcium overload in the cardiomyocytes, and it leads to the activation of calpain, and this can explain the reperfusion injury. Calpain specific inhibitor is called calpastatin, and it is involved in the proteolysis of the amyloid precursor protein. The calpastatin system is involved in membrane fusion events. This shows approximately the different forms of calcium in iron. So you can see the ionized form and the complex with anions such as bicarbonate and citrate, and this column, this 5+1, is exchangeable and diffusible. The ionized form and the complex with anions it's diffusible, whereas the bound with proteins, mainly with albumin, fraction is non-diffusible. Factors regulating blood calcium level are mainly vitamin D, PTH, and calcitonin. I shall talk to you briefly about PTH and vitamin D. Calcitonin will be dealt in another lecture. So PTH, it is secreted by the 4 parathyroid glands. Chief cells secrete the PTH. It is synthesized as pre-pro-PTH with 115 amino acids, and in the endoplasmic reticulum and Golgi apparatus, this is cleaved to mature PTH with 84 amino acids. and the storage is only for about an hour. First 35 amino acids are active. These are the biologically active components of the PTH, and it is released by negative feedback by ionized calcium in serum. As the serum calcium increases, the PTH release decreases, and the release is mediated by cyclic AMP. Normal PTH levels are between 10 to 60 nanogram per liter, and in primary hyperparathyroidism it increases to 100 nanogram per liter. And this is the mechanism of action of PTH. It binds with the receptor protein on the surface of the cells. This activates adenyl cyclase. This increases calcium in cells. And there are three major sites of action: bone, kidney, and intestines. And in all these three sites, it increases the serum calcium level. What does it do to the bones? It mainly demineralizes. It induces pyrophosphatase in osteoclasts. The number of osteoclasts are also increased, and these stimulated osteoclasts release lactate into surrounding medium, which makes it acidic, and it solubilizes the calcium. It also causes secretion of collagenase from osteoclasts, so it will result in matrix dissolution, and mucopolysaccharides and hydroxyproline are excreted in urine. But this being said, this is what we know from our medical school. PTH also is supposed to increase the bone mineral density and the bone mass by another mechanism that will be dealt with in another lecture, where I will explain how that does, because PTH, when given continuously, could cause the demineralization, but when intermittently, it could increase the bone mineral density. PTH, the action on kidneys and GIT. PTH in the kidneys will cause decreased renal excretion of calcium and increased excretion of phosphate, whereas vitamin D will decrease both decrease the renal excretion of both calcium and phosphate. This action is mainly through increase in reabsorption of calcium from the kidney tubules. In the intestines, it will stimulate 1-hydroxylation of 25-hydroxycalciferol. PTH stimulates 1-hydroxylation of 25-hydroxycalciferol in the kidneys to produce calcitriol. This calcitriol will increase calcium absorption from intestine. So PTH via calcitriol indirectly increases calcium absorption. Vitamin D also will influence the calcium absorption. So cholecalciferol is synthesized from 7-dehydrocholesterol in skin under the influence of sunlight. It is hydroxylated 25th position in liver and at first position in the kidney. The active derivative is called the dihydroxycholecalciferol or calcitriol. Vitamin D, how it increases absorption of calcium from the gut. So as you can see that calcitriol enters the cell and it binds with the receptor, and the calcitriol receptor complex now moves to the nucleus, and then it interacts with the DNA, and it causes de-repression and consequent transcription of specific genes that code for calbindin. And calbindin will cause, it's a calcium binding protein, and it will cause increased absorption of calcium. This is how vitamin D will increase the calcium absorption in the gut. And it also increases the phosphate absorption also. And in the renal tubules, calcitriol increases the reabsorption of calcium and phosphorus by renal tubules, and hence more minerals are conserved. But you have to remember that PTH only will cause the reabsorption of calcium, and it will increase the excretion of phosphate. Calcitriol will increase the secretion of clotho-protein from the kidney, and this in turn causes increased expression of the epithelial calcium channel, also called TRPV5, and this leads to increased reabsorption from the distal convoluted tubule. So clotho also increases intestinal calcium absorption by increasing expression of another calcium channel called the TRPV6 in the intestinal cells. and this protein has beta glucosidase activity, and clotho is produced predominantly in the kidney. Clotho deficiency will result in increased production of vitamin D, and variants of clotho gene are associated with human aging. It may also protect the cardiovascular system through nitric oxide production. So this is a chart which compares the three major factors affecting serum calcium. Calcitonin, I will be discussing it in the next lecture. So in the blood calcium, vitamin D and PTH will increase, and PTH will drastically increase the blood calcium, whereas calcitonin will decrease it. And calcium absorption is mainly increased from the gut in case of vitamin D, whereas PTH causes demineralization, and calcitonin opposes demineralization. Calcium absorption from the gut, vitamin D and PTH increases. Bone resorption, vitamin D decreases and calcitonin decreases, whereas the PTH increases. And vitamin D deficiency results in rickets. It doesn't show any forms of tetany, whereas PTH deficiency will result in hypocalcemia, and it will show the tetany. In rickets, vitamin D is the drug of choice. PTH is contraindicated in rickets, and calcitonin could be beneficial. So excess vitamin D could produce hypercalcemia. Excess PTH could cause marked hypercalcemia, whereas calcitonin, if there is an excess, it can produce hypocalcemia. The relationship between phosphorus and calcium. There is a reciprocal relationship of calcium and phosphorus. The ionic product of calcium and phosphorus in serum is kept at a constant. So it's around 40. In renal insufficiency, phosphorus excretion is diminished, and phosphorus levels increase in blood, and calcium is lowered, and this could lead to tetany in the renal failure. Relationship between serum proteins. In hypoalbuminemia, total calcium is decreased. This because calcium, around 40% of the calcium is bound with the albumin. So for each gram of albumin lowered, about 0.8 milligram of calcium is reduced. But there is the metabolically active ionized form intact, so there will be no deficiency manifestations even though the whole calcium is decreased. Alkalosis and acidosis. Alkalosis favors binding of more calcium with proteins, with consequent lowering of ionized calcium. So in respiratory alkalosis, when there is hyperventilation, you could see features of hypocalcemia even though the total calcium may be normal. So in alkalosis, even though the total calcium is normal, the ionized calcium will go down. Hence the calcium deficiency may be manifested. And acidosis favors ionization of calcium. Renal threshold for calcium is around 10 milligram per deciliter, and when this level is reached, calcium is excreted. And when injected intravenously, most of the calcium is rapidly excreted. Now coming to phosphate, the total body phosphate is around 1 kilogram, and 80% is seen in the bone and teeth and 10% in muscles. It is mainly an intracellular ion, and it is seen in all the cells. About 500 milligram per day is required. Milk is a good source. Other sources are cereals, nuts and meat. And vitamin D will increase the phosphate absorption. Serum levels are around 3 to 4 milligram per deciliter in normal adults, and it is slightly more in children. Fasting levels are higher. The postprandial decrease is due to the utilization of phosphate for metabolism. And monovalent and divalent phosphate ions, they are present in the ratio of 1:9 in alkalosis in the plasma, and at a ratio of 1:4 when the pH is normal. The whole blood phosphate is 40 milligram per deciliter. This is almost 10 times more than the serum level. This is because WBCs and RBCs contain a lot of phosphate. So hemolysis should be prevented when blood is taken for phosphate estimation. And phosphate regulation, as I've told you before, in the case of calcium, it holds an inverse relationship with calcium. Excess of serum calcium or phosphate results in excretion of the other by kidney. The phosphate level is regulated by excretion through urine. The renal threshold is 2 mg per deciliter, and usually 500 mg of phosphate is excreted through urine per day. There is a diurnal variation with the highest output occurring in the afternoon. And PTH increases calcium and phosphate released from the bone and decreases loss of calcium, that is the renal excretion of calcium, and increase urinary loss of phosphate. So these are the cause of hyperphosphatemia. It could be from increased absorption, it could be due to increased cell lysis, or it could be due to decreased excretion of phosphorus. Other causes are hypocalcemia, massive blood transfusion, thyrotoxicosis, and drugs. So increased absorption takes place when there is excessive vitamin D, or if you give phosphate as an IV infusion. Cell lysis occurs when there is chemotherapy for cancer or bone secondaries, and in rhabdomyolysis. And decreased excretion occurs in renal impairment, hypoparathyroidism, etc. And drugs, especially nifedipine and furosemide, can cause hyperphosphatemia. Hyperphosphatemia could be caused due to decreased absorption of the phosphate. So as in malnutrition, malabsorption, chronic diarrhea or vitamin D deficiency, or when there is an intracellular shift such as in insulin therapy, respiratory alkalosis, etc. When there is increased urinary excretion of phosphate such as in Fanconi syndrome, hyperparathyroidism, or hyperphosphatemic rickets. And it could also be due to chronic alcoholism, which is an important cause, or hypercalcemia of any cause. As I've told, calcium and phosphate has got an inverse relationship, or any case of hereditary hypophosphatemia. Rarely drugs also cause this, like antacids, salicylates, diuretics, etc. Now coming to magnesium, it is the second most prevalent intracellular cation, and it's mainly seen in the ICF. Total body magnesium is around 25 gram, and 60% is complex with calcium in the bone. 1/3 of the skeletal magnesium is exchangeable with serum. And magnesium, if given orally, produces diarrhea usually, but if it is given IV, it produces CNS depression. Requirements is about 400 mg per day for men and 300 mg per day for women. And if you give more than 600 mg per day, it may cause diarrhea. And the requirement increase during lactation. Normal levels are between 1.8 to 2.22 mg per deciliter, and if it is less than 1.7, you will have hypermagnesemia. Inside the RBC, the magnesium content is 5 milliequivalent per liter, and in the muscle tissue you can see that it increases even more. It's around 20 milliequivalent per liter. Around 70% is in the free state and 30% is protein bound. Serum must be separated from the clot as soon as possible because hemolysis can increase the magnesium levels. And magnesium is regulated by intestinal absorption and by renal excretion. It is reabsorbed from the loop of Henle only, and it is not from the proximal tubules. Hypomagnesemia is caused by the deficiency of magnesium, and it leads to neuromuscular hyperirritability, whereas increased magnesium will cause neuromuscular or neurological depression. So deficiency will cause irritability of the CNS and also cardiac arrhythmias. And the magnesium deficiency symptoms could be similar to that of calcium deficiency, but the symptoms will be relieved only when the magnesium is given. Acute symptomatic deficiency is treated by giving parenteral magnesium. Oral therapy may lead to diarrhea, so intravenous magnesium sulfate is preferred in most acute scenarios. These are the relationship with calcium and potassium in hypomagnesemia. When there is these are the effects on calcium and potassium when there is hypomagnesemia. When there is magnesium deficiency, PTH secretion is inhibited and peripheral action of PTH is attenuated. So hypomagnesemia will induce hypocalcemia, and this hypocalcemia will not respond to calcium treatment unless the magnesium deficiency is corrected first. Hypokalemia is also frequently seen in association with hypomagnesemia, and even if you try to treat this with potassium, it will be not successful unless the magnesium is replaced at the same time. So whenever you have hypokalemia and hypocalcemia, magnesium should be checked, and if magnesium is deficient, you need to replace the magnesium first. So causes of hypomagnesemia are if there is increased urine loss, such as in tubular necrosis, hyperaldosteronism, familial causes of hypomagnesemia, increased intestinal loss due to diarrhea, laxatives, ulcerative colitis, suction or vomiting, and then in liver cirrhosis, malabsorption, protein calorie malnutrition, hypoparathyroidism, and toxemia of pregnancy. Also, drugs like thiazides, aminoglycosides, cisplatin, haloperidol, alcohol, even PPIs could produce hypomagnesemia. When to test for magnesium? When there are cardiac arrhythmias, when there is resistant hypokalemia, and also tetany not responding to calcium therapy. That means the hypocalcemia is induced probably by the hypomagnesemia, and also in pregnancy with preeclampsia. Hypermagnesemia, on the other hand, is uncommon and often due to excessive intake. And magnesium intoxication causes depression of neuromuscular system, causing lethargy, hypotension, and also it causes respiratory depression. And it will result in bradycardia and weak tendon reflexes. In severe cases, acute rhabdomyolysis could result. And hypermagnesemia also causes decrease in serum calcium by inhibiting PTH secretion, which in turn will have deleterious effects. And causes of hypermagnesemia are if there is excess intake, renal failure, hyperparathyroidism, oxalate poisoning, rickets, dehydration, multiple myeloma, etc. And aminoglycosides can cause hypermagnesemia, as well as antacids containing magnesium, also calcitriol, etc. Thank you.